CAR T-cell therapy is probably at the beginning of a paradigm shift on how we might be treating B-cell malignancies as well as other lymphomas, including Hodgkin lymphoma and T-cell lymphoma in the future. It's further along in B-cell malignancies. It's now been approved in at least 2 different lymphoma subtypes, aggressive B-cell lymphoma as well as mantle cell lymphoma after two or more lines of therapy in the case of aggressive B-cell lymphoma, and after 1 or more lines of therapy in the case of mantle cell lymphoma. And approval is also expected for indolent B-cell lymphoma after at least 2 or more lines of therapy. The appealing thing about this therapy is that it's a one-time infusion, and it can induce long-term remission and probably cures in a subset of these patients even after failing standard chemotherapy as well as stem cell transplant. So I think it makes sense to move this to earlier lines of therapy to see if we can further improve the efficacy of this regimen and decrease the morbidity and mortality related to multiple treatments in these patients. But we also have a lot of work to do. If we look at aggressive B-cell lymphoma, we see that about 60% of these patients still relapse after CAR T-cell therapy in the third-line setting. And if we look at patterns of relapses to see why they relapse, about a third of these patients seem to have antigen loss with respect to axi-cell treatment, and 2/3 of the patients have CD19 positive relapses. And if they have antigen loss, it would make sense to target other B-cell targets with CAR T-cell therapy. So now CAR T-cell therapy against CD20 as well as CD22 and BAFF receptor and CD79B are currently in various stages of preclinical and early clinical development and are being tested in the clinic. And in fact, bispecific CAR T-cells are also being evaluated targeting CD19 and CD20 or CD19 and CD22 in early stage trials. That could potentially improve the efficacy even further. In the other 2/3 of the patients, we see that a major reason for relapse is probably because of dysfunctional T cells, which could be at the time of the apheresis, the T cells could be dysfunctional because these T cells have been affected either due to the chemotherapy itself or the disease biology itself. Or sometimes the T cells could be dysfunctional after they have been infused when they reach the tumor microenvironment because of various immune checkpoints in the tumor microenvironment. So there are various strategies that are being explored to address this impaired T cell fitness or function. One strategy is to use CAR T cell therapy in combination with immune checkpoint blockade or other immunomodulatory agents such as lenalidomide to further improve the efficacy of CAR T cell therapy. Another strategy is to use generate CAR T cells from a healthy donor and use allogeneic off-the-shelf CAR T-cell therapy approach. And allogeneic CAR T-cell therapy approach has some appeal also because as opposed to autologous CAR T-cell therapy where each manufacturing run results in only one product, with an allogeneic CAR T-cell therapy approach from each manufacturing run, one could potentially generate and treat at least 100 patients at a time, so which could potentially decrease the cost of this approach. So allogeneic CAR T-cell therapies are also in early stage clinical trials. And at ASCO this year, the results from the phase 1 study of ALLO-501, which is an allogeneic anti-CD19 CAR T-cell therapy, were presented. This is the ALPHA study where the anti-CD19 CAR T-cell therapy is generated from a healthy donor, and in order to prevent graft-versus-host disease, the T-cell receptor is knocked out or genetically edited by using the TALEN technology. In addition, the CD52 molecule on the T-cells is also knocked out by gene editing to allow usage of anti-CD52 monoclonal antibody in combination with Cy/Flu as part of the conditioning regimen to prolong the lymphodepletion in the host and allow increased persistence of the allogeneic CAR T cells. So this was early data from this study was presented with, where 22 patients have been treated with relapsed or refractory large B-cell lymphoma or follicular lymphoma. And this data shows that this allogeneic CAR T-cell therapy is quite safe. Only a third of the patients had CRS with mostly grade 1 and grade 2. There was no neurological toxicity that was observed. There was no graft-versus-host disease that was observed in these patients. And in terms of efficacy, at least the response rates look quite comparable to what we see with autologous CAR T-cell therapy, with an overall response rate of 63% and a complete response rate of 37%. And it looked like these response rates could be optimized by optimizing the conditioning regimen. For example, in the patients who received a higher dose of anti-CD52 antibody conditioning along with Cy/Flu, they had a complete response rate of 50% as compared to the low dose anti-CD52 conditioning, where the complete response rate was only 27%. So I think these data suggest that allogeneic CAR T-cell therapy looks promising in lymphomas, at least in terms of the safety and the short-term efficacy seem quite comparable or maybe even better compared to autologous CAR T-cell therapy, but longer follow-up is needed to determine whether those responses would be durable.